Alrighty, starting the recording for week 10 here. And jump off with DNA. So we're going to be talking all about DNA. Now, DNA, deoxyribonucleic acid, is the molecular basis for inheritance. Now, let's break that down into a few pieces. So when we say molecular, usually we're talking about things at the molecular level, at the atomic level. So structures of atoms is where we're looking right now. That usually involves DNA, RNA, proteins, enzymatic activities, things at that level. So that's the level that we're looking at, and then going further, we see it's the basis of inheritance. It's it's how traits are passed from one generation to the next. So this is the molecular level where we're looking at how traits are passed. So Watson and Crick were two very early scientists that were working on the structure of DNA. This was sort of a race at the time, is who would be the first to describe the well, first it was a race to see who could figure out how traits are passed. Eventually, they found out that DNA and RNA were the information. Then the race came to determine the structure of DNA. What does it actually look like? Let's create a model of it. So that was Watson and Crick sort of are renowned for completing that first. Much like Darwin was the first to accurately describe a mechanic for evolution to occur. But yes, moving on. So what is DNA? DNA is hereditary information and is reproduced in all cells of the body. So every single cell in your body is going through this unless it has already died. Skin cells are an example where skin cells at the surface, they're not doing much of anything. But when you go underneath, all the skin cells underneath are still very much going through this constant process of DNA replication. Anytime a cell in your body splits, anytime it goes through meiosis or mitosis, and remember, all your body cells will be going through mitosis. All your somatic cells will be going through mitosis. Anytime that happens, we have to go through DNA replication first. And DNA is responsible for a large portion of what makes an individual organism the way they are. We know now that DNA isn't everything, that environment plays a factor, and also epigenetics, so there's protein markers on the DNA itself that aren't coded into it but are attached to it that might have effects. So there's a number of other things, but DNA is by and large the biggest contributor to what makes an organism the way it is. And here's a quick little picture of the structure that we attribute to DNA nowadays, the double helix. So what I mean by double helix is we have, if you follow one strand all the way down, it is a single helix, and they are running parallel to one another, so it forms a double helix. Here is a photograph of Watson and Crick next to a model for DNA. So DNA is copied during DNA replication. So that's when we actually go through the process of making a copy, is during the DNA replication phase. And cells can repair this DNA. Anytime you're copying something, it's not going to be perfect. So there's tons of DNAs that help to fix, pretty much proofread the copies of DNA that are being replicated. So in the early 1900s, so early in the 20th century, the identification of molecules of inheritance is what they referred to them, became sort of the main focus of the age for biology. And there was all sorts of theories that were proposed before we figured out that DNA was the molecule responsible. Yeah, there was several virus studies that looked at viruses and bacteria and the different components of bacteria and viruses to figure out which particles actually transmit DNA. The first evidence of this was a clever little experiment by Frederick Griffith, who took 2 strains of bacteria. So a strain is sort of like a smaller group within a species for bacteria. So it's not quite a different species, but we don't have things like reproductive isolation because they're asexual, so we just say that's a different strain. But he had one strain that was pathogenic and one that was harmless. He killed one with heat, the pathogenic one with heat, and then introduced the non-pathogenic strain live and determined that they must have gotten some kind of characteristic that made them become dangerous. So we had one dangerous strain, one harmless strain, and just being near the leftover materials of the harmful strain allowed the normally harmless strain to then be dangerous. He called the process transformation, and this is now what we use to describe any time an organism assimilates DNA into its genome. Anytime you take up DNA and use DNA from an outside source, that is called transformation. And bacteria still do this all the time. So here is a mock-up of what that experiment looked like. So we have our control is where we take either just the pathogenic, where the mouse dies, or just the non-pathogenic, where the mouse is healthy. So this is the harmful strain. This is the more innocent strain. Then we are going to heat kill the pathogenic strain, and we show that there's no disease then. So they've taken the pathogen and raised it up to a high enough temperature to where those cell membranes dissolve. For all intents and purposes, the bacteria is dead, and we inject that into the mouse, and it's fine. It's healthy. But when we mix the remnants of those, that's where we get the uptake of the harmful bacteria's DNA into the non-harmful bacteria, and then they express those genes and become deadly. All right, so there was several other experiments that used viruses that kill bacteria and then the bacteria themselves to determine that the RNA or DNA inside of that protein coat is their instructions. That's their genetic instruction. All right. It was Alfred Hershey and Martha Chase that ran that experiment. And it is a fairly complicated experiment, so we're not going to really go into it in here, but there is more about that in your textbook if you'd like to look into it. So once the structure of DNA was starting to be described, we also determined, or the presence of DNA as the genetic molecule. So by the 1950s, we knew that it was the genetic molecule through those experiments. They were able to describe 4 different types of bases, essentially, of letters. So those are the nucleotides, or those ended up being the nucleotides that we know about now. But they were just figuring out what the content of the DNA was at the time. Basically, what the scientists came up with was that certain base pairs are only connected to certain base pairs. And it wasn't until we got the double helix that we finally understood why that was happening. So here is a chart of those base pairs. This is a good one to, no, maybe not store in the old memory bank, because this is a lot of information here, but know that adenine and thymine go together. So across from this thymine is an adenine, across from this guanine, or across from this guanine over here will be a cytosine, across from this cytosine will be a guanosine, across from this adenine will be a thymine. And so going across here, we have the nitrogenous bases. Over here, we have the nucleotides. So that is a sugar and a phosphate and a sugar and a phosphate and a sugar and a phosphate, sugar, phosphate. After or off of every sugar, we have a nitrogenous base. And which end has the phosphate hanging off when we look at the pair of across this double helix? If we look at the pair, which one has the phosphate hanging off is how we are assigning this 5' end and this 3' end. Yeah, that's the base. This is one strand of DNA, by the way. This is not a double strand of DNA, because this is showing each of these, essentially, this thymine is one base pair, and then across from it would be another one over here. All right. So here is sort of the structural difference of DNA and RNA. Now I can't play the video in this recording, but if you go and look at the slides, it is available on the slides. Just know that RNA is single-stranded and DNA is double-stranded. Although there is certain situations where the RNA can in places fold on itself and bind. All right, so after DNA was accepted as a genetic material and we started to learn more and more about DNA, there was a race to determine its structure. Famously, Watson and Crick were the first, were considered the first to publish. But one thing that is sometimes neglected in that is that Maurice Wilkins and Rosalind Franklin were studying X-ray crystallography, or were filming in X-ray crystallography. They were using X-ray crystallography to capture images of DNA that had never been studied before. And they the pictures that they used were later used by Watson and Crick without their knowledge. So here is Rosalind Franklin, and that is the picture that she was able to take of DNA. So out of this picture is, well, this picture is one of the main pieces of evidence that Watson and Crick used to develop their model for the double helical structure of DNA. All right. So without her X-ray crystallography pictures, Watson and Crick would not have been able to come up with their model, or at least not nearly the same amount of time they did. Someone else might have beaten them to it. Particularly, Franklin herself may have beaten them to it. But yeah, they were able to determine how the base pairs matched up with one another in a 3D structure using the image. And many calculations. All right. So here, in that previous picture, we were just looking at this one side of the DNA molecule. So now when we look at it in its a more complete form, we have the matching base pair, the complementary base pairs. So T to A, G to C, C to G, A to T. And the 3D structure, when it folds, will line up like this. And keep in mind that because like how these bases are hooked up to each other is different depending on which strand because they are running anti-parallel to each other. Remember, we have the phosphate end on one side. One strand has it on one side, one strand will have it on the other. So the free phosphate end is over here, free phosphate end for this other strand is over here. They're always opposite like that. Again, there is another animation that can be viewed in the slides that I cannot play here. And just sort of keeping in mind the structure of DNA. So the blue ribbon here represents the sugar phosphate backbone, that interlinking sugar phosphate backbone. And we can see that it doesn't match up sort of like perfectly symmetrically. It's sort of offset a bit. So it's sort of like a ladder that's been folded around a bit. All right. So again, the term for the 5' and 3' end, you might have heard those terms before. The term for them is they run anti-parallel, and as we can see, they are always opposite on either end. 5' ends are opposite, 3' ends are opposite. All right. We also determined with this that the reason why we have this AG and or AT CG pairing, A's with T's, C's with G's, is because of the 3D structure. So think of this being part of that double spiral. In a twisted double spiral, it won't fit if you have the smaller pyrimidines both lined up with one another. It won't fit if you have two of the larger purines lined up with one another. But if you have one of each, they line up well with the long strand that is forming. So there is a physical deformity that happens where these two won't be able to reach each other, or these two are bulging and pushing each other out if there is a coding error that happens during DNA replication, which we'll be talking about more next lecture. All right, Watson and Crick reasoned that pairing was more specifically dictated by like the structures, those physical structures. So only A and T could only stably pair with one another. G and C could only stably pair with one another. Here's a look at what those bonds look like. So A and T, we see uh, on the left is always going to be the sort of larger structure here, the purines. And on the rights, we have the pyrimidines. So thymine and cytosine are our pyrimidines. And between A and T, between these two pyrimidine and purine combos, we have 2 hydrogen bonds, whereas between guanine and cytosine, we have 3. So there's no way for a cytosine to pair up with an adenine because there's an inappropriate number of bonds, even though the 3D structure might work out for the sugar phosphate backbone. They won't sort of fit together because of these hydrogen bonds. And that can be true for any of them. So effectively, what nature's given us here is a code. by which we always have one going with another, so C is always going with G, and anytime we have a differentiation in that code, we have the ability to create a large amount of information. So there's a lot of proteins that get to work with DNA replication. We focus on a few choice ones in this class because it can get a little bit overwhelming if you look at just the number of proteins. But yeah. All right. So after they determine the structure, they try to determine the mechanism for replication. So how does this molecule get reproduced? So they knew that we had these two strands now. How do we make new strands? Every time they split, we have more. So there's something going on that is replicating them. And there was a few strategies that were sort of proposed. The one that ended up having the most credibility and was later verified was the semi-conservative model. So semi means half, conservative means saved model. So half of our DNA is saved or old. So here is what a conservative model would look like is we are conserving one entire copy and then just making a new cloned copy. A dispersive model would be where different pieces of each end up going into new copies. So if this was happening sort of more scattershot, but what we end up seeing is the two strands separate, which is the semi-conservative model. Those two strands separate, and then replication happens on each strand, and they continue to sort of unzip and replicate out from there. All right, we're going to go more into DNA replication starting next class. But what I want you guys to do is I have a video posted for our Turn and Talk. It is about Rosalind Franklin. And it's like a 5-minute TED Ed video on her and sort of her story. And I want you guys to use that video as context to sort of launch a discussion, which is, while we've come a long way since Rosalind Franklin's time in addressing some of the problems with sexism, we still have a long way to go. Where do you think we have improved in terms of sexism, either in the workplace or around science in particular? What's still broken? And what can we do to help? I would really love to know your guys' thoughts and opinions on this. But yeah, that is going to be available on the discussion forum. For next class, we're going to be reading more into chapter 16. Specifically, we're going to be going into the section on DNA replication. With that, I hope you guys have a great rest of your day, and I will see you all next time.